Hello, my name is Satoshi, and I'll be your host for today. Welcome and thank you for taking part in MDPI's webinar entitled Glioblastoma, Intratumoral Heterogeneity and Plasticity. This is the first webinar in the International Journal of Molecular Science series, based on a special issue from our journal of the same name. If you are interested in contributing to this topic, don't hesitate to submit your research. Simply check the webinar's webpage for more information. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's webpage. Today's session is also being live streamed on MDPI's YouTube channel. So hello to those of you watching us on there. Certificates of attendance are available upon request. To get yours, please send an email to ijms.webinar@mdpi.com. ijms.webinar@mdpi.com. Certificates will be delivered to those who have registered and are watching on Zoom, not on YouTube. Thank you for your understanding. There will be 3 presentations today of 30 minutes each. The presentations are followed by a question time moderated by the chair. If you have any questions for our speakers, please post them in the Q&A box at the bottom of the screen. Please remember to mention the name of the person to whom you are addressing your question. Today's webinar is chaired by Dr. Guillermo Gomez from SA Pathology and the University of South Australia. Dr. Guillermo Gomez is a rising star of the field, and he has conducted unique cross-disciplinary researches in organoid biology, mechanobiology, single-cell transcriptomics, clinical oncology, and artificial intelligence as a group leader of the Center for Cancer Biology at the University of South Australia since 2017. He published more than 40 papers in leading journals in the last 6 years. Career-wide, he has published 70 papers and total citations are over 3,200. Thank you, Guillermo, for chairing today's webinar. That's it for me. I now hand over the microphone to our chair. I hope you all enjoy.
Thank you very much, Satoshi. Welcome everyone to this special webinar on glioblastoma intratumoral heterogeneity and plasticity. And I hope all of you enjoy it. Just a quick announce. So this will be the first seminar. We're also planning a second seminar later in July, but just keep watching this channel. Okay, that is on recent work that we have done in the lab about harnessing artificial intelligence to understand intratumoral heterogeneity in glioblastoma. So glioblastoma is the most aggressive type of brain cancer. It's a very heterogeneous. It has a survival, 5-year survival less than 5%, and its clinical management hasn't improved in the last 30 years. So that has been very challenging in the field. So currently the standard of care for glioblastoma is the maximal safe surgical rejection, chemoradiotherapy. However, the tumors inevitably recur. And for the cases of recurrence glioblastoma, there is no available treatment. So this highlights the urgent need for new and better therapies for glioblastoma. So, as I mentioned before, glioblastomas are very heterogeneous, and this has been early seen in the histopathology, where the phenotypes or how the glioblastomas are shown in the images varies widely between patient to patient. Also, they don't occur homogeneously in different brain regions. So, the frontal lobe is the region, the brain tumor region where more often occur the glioblastoma, but also this type of tumors occurs in other parts of the brain. At the cellular level, when we look at a single tumor, for example, using single-cell RNA sequencing, we can see that these tumors not only contain tumor cells, but also they are quite infiltrated by non-malignant cells that are co-opted by the tumor cells to allow tumor progression. So what we have seen, Maria Jose and Brian will talk also, is that these tumors contain, for example, tumor-associated macrophages, brain-resident macrophages like microglia, endothelial cells, pericytes, because these tumors are also highly vascularized, and lymphocytes. Also, tumor cells are not equally across a single tumor. Recent data have shown that tumor cells are highly plastic and present mainly into 4 different transcriptional states, described like an oligodendrocyte precursor state, astrocyte-like, neuronal precursor state, and mesenchymal-like state. So the plasticity of these tumor cells is conferred by their stem cell properties, and the proportion of these cells depend on their close interactions with non-malignant cells in the tumor microenvironment. So this high level of intertumor heterogeneity in glioblastoma is one of the main challenges to develop new therapies. And a lot of research now is trying to focus on that particular topic, and that is the focus also of the special issue in the IGMS that we organize. So to continue with the presentation, we just now go to some early work that we did before, trying to understand more about this heterogeneity that occurs in glioblastoma. So one of the things that we have used is artificial intelligence to analyze glioblastoma histopathological images. So in the first attempt to do this, so this is something. In the first attempt that we did, we classified the survival rates of glioblastoma slides of glioblastoma patients based on the histopathological images. So basically, we classify. We use the histopathological images from patients that have different survivals, and using AI, we were able to predict how long a patient is going to survive. So these early results show us that there are information in the slide that is meaningful in order to understand the biology and the clinical outcome of the patients. So later we wanted to use AI to know and identify what are the image features that relate to glioblastoma intertumor heterogeneity that contribute to poor survival in GBM. And also we wanted to use this to generate a spatial atlas of glioblastoma that can tell us about the cellular composition in different tumor regions and different type of interaction that occurs between tumor cells and non-tumor cells. So this is a recent work that we published in the British Journal of Cancer last month, where we use AI to segment the tumor regions, the central tumor microvascular proliferation region, the infiltrated region, and the leading edge region from histopathological slides. The work was driven by a PhD student in the lab, Amin Sadeh Shirazi, and he's finishing his thesis at the moment. So to implement AI for this analysis, we set out the following pipeline, where at the beginning we used the IBGAP data set to train a deep convolutional neural network model. And then basically we test different options of architectures and interparameter tuning to train the model and to verify optimal results. So in this first phase, as I mentioned, we use the IBGAP data set where we have histopathological images and annotated tumor regions. These In IBGAP, we have 32 patients. Those were corresponded to 805 whole slide images. And then for training of the model, we'll use 687 slides from 28 patients. For validation, we use 52 slides from 2 patients. And for testing, we use 48 slides from 2 patients. So these are, we tested 52 different models that were trained, validated, and tested. From there, we pick up one that leads to the better training results, and that's the one that we choose. So these are the results we use in the IBGAP dataset, where we can see that segmentation pattern delivered by the model was even better than the ground truth that we obtained from the IBGAP database. Then we apply this model to TCGA. TCGA is the Cancer Genome Atlas and contains data from 329 patients with glioblastoma and the corresponding 640 whole slide images. Using AI, we were able to segment this very rapidly in a period of 2 months, approximately, once we have the training model. And the advantage of using TCGA is that we also have much RNA sequencing data. So this slide summarizes the power of artificial intelligence for this project, because we were able to assess very rapidly, segment the different tumor regions in a large number of histopathological slides, which by human means might have taken a lot of time, in the order of a year or so. So we then quantify the size of the different tumor regions across the different slides in the TCGA database, and this is the distribution of the findings. Most of the slide contains regions that correspond to central tumor necrotic area. That makes sense, because this is the part that basically is more highly resected by the neurosurgeon, right? So then we perform correlation analysis for different groups of patients with the TCGA. TCGA has a notation for the kind of mutation that each sample contains, and then we perform correlation between survival and tumor region, the presence of tumor regions for different patients with different mutations, right? And what we found is that there was a negative correlation between the presence of lineage and infiltrated tumor, and also the presence of microvascular proliferation for this type of mutation, for EGFR mutation and NF1 and PTEN and TRAP. And that is very indicative of the aggressiveness that this type of tumor has when they are infiltrate the surrounding parenchyma. So we then wanted to go a step further to better understand, these tumor regions and what kind of transcriptional programs they activate. So in order to identify genetic signatures associated to each tumor region, we then perform experiment correlation analysis between the size of a tumor region segmented by artificial intelligence for the use of our model, and the amount of the transcripts that are expressed by the gene across the different patient samples. So in red are genes that are positively correlated, and blue are negatively correlated. We found that different tumor regions contains different number of genes, but interestingly, the infiltrated region and the microvascular proliferation region has a significant number of genes which expression is negatively correlated with survival. So in infiltrated tumor, we got around 180 genes negatively correlated with survival, and the central tumor microvascular proliferation we have around 324 genes that are negatively correlated with survival. We took a look more closely to the microvascular proliferation region, and this is we choose this region because one is the large number of genes that are especially correlated with this tumor region, but also because the presence of vasculature or the high level of vascularization of glioblastoma is a key characteristic of its aggressiveness. So when we run the gene ontology analysis for cellular processes for this genetic signature, we found that this is highly correlated with the immune system processes, the immune response, and also a lot of secretory mechanisms, like a secretory by cells, a basically mediated transport, extracellular matrix, and cell motility that correlates with also with the invasion of phenotype of glioblastoma. We then wanted to analyze what is the cellular composition of these different tumor regions based on the genetic signatures that we identify. And for that, we use CellKV, that is a database that correlates, trying to deconvolve expression patterns using single-cell RNA sequencing data. And we found that genetic signature for the infiltrated tumor region and the central tumor microvascular proliferation region are expressed mainly by non-malignant cells in the tumor microenvironment. For example, in the infiltrated tumor region, the signature was specifically expressed by microglia and monocyte cells, and the central tumor microvascular proliferation region was expressed by endothelial cells, microglial cells, macrophages, and monocytes. We also then wanted to verify these results using single-cell RNA sequencing data derived from patient biopsy that we collect here in Adelaide. And we found a nice correlation with the in silico results. So here we analyzed this, again, the central tumor microvascular proliferation gene signature, and we found that many genes, here are the genes of the signatures, and here are the cells that express those genes. And we found that here a cluster of genes are expressed mostly by microglia and macrophages, here by endothelial cells, and here by pericytes that are important in forming the blood-brain barrier. So, also, there are genes that are specifically expressed by tumor cells. So then we have a more deep picture about the cellular composition that occurs in different tumor niches. We wanted to take a look what are the potential interactions that might occur between tumor cells and non-malignant cells. And here we took a simplistic approach just to start the process of what is characterizing these interactions, and was to identify ligand receptor interactions that are expressed by pairs of non-malignant cells and tumor cells. We perform an in silico analysis using our single-cell RNA sequencing data, and we found that a pair of reticulum 4 ligand and a receptor GJB2 that is a G protein coupler receptor is expressed across the different patients samples that we analyze. And this ligand receptor pair also correlates significantly with survival of patients. For example, a patient that has low expression or high expression of reticulum 4, the survival rates are clearly different, with those that express more receptor having a shorter survival. More of the ligand, this reticulum 4, has a shorter survival. The same for the receptor. So overall, this approach permits us to have a better understanding of the cellular composition of the tumor microenvironment, and also the genetic signatures that are expressed by the different cells, and eventually a better understanding of possible tumor non-malignant cell interactions that are important in driving tumor progression. So the summary of the AR results is that it has been very powerful to segment this large number of histopathological images into different tumor regions, and being able to take advantage, of already available databases to get these signatures, right? That are also important in survival. So with this, we've been able to identify, new interaction that can serve as a potential targets. And now I will go to the next part of the talk, where I will just focus a little bit on how we can proceed from our in silico data to a more functional test of all findings, right? So I will just mention briefly what we have done. Brian will go in deeper detail later in his talk as well. But overall, there was a revolution in all the field of cancers with new in vitro models for preclinical tests of drugs. In glioblastoma, there are probably different ones, but two key important models are those ones that relate to the culture of patient-derived glioblastoma stem cells, and also the culture of patient-derived glioblastoma organoids. They have a different advantage. So the 2D cultures maintain the stem cell properties of stem cells, are maintained serum-free media, and are undergone, and those are important when lower passages, when used at lower passages. Whereas the 3D organoids, these recent technologies developed by Jacob et al. And published last year, are derived directly from the tumor biopsy, are generated within a 2-4 weeks time period. And the important thing is that these organoids contain different cells in the tumor microenvironment as well. So contain tumor cells and non-malignant cells. So we have a program set up here in South Australia, where we established collaborations with neurosurgeons and neuropathology in the hospitals, and that is linked to South Australian Neurosurgical Tumor Bank, that also is linked nationally to other banks and hospitals, including the one that is in Queensland with Brian as well, where we can access fresh patient tumor tissue and blood. And here in Center for Cancer Biology, we collaborate with Stuart Phipson, Michael Baum, Tessa Gargett, and Lisa Eber to generate different living glioblastoma biobank that contains blood, generate glioblastoma cell lines, and generate tumor organoid explant culture. Recently, we developed a screening pipeline where we can test the effects of the functionality of new targets that we identify. And the pipeline consists of high throughput screening using the cell lines to rapidly identify drugs that can kill tumor cells. But then we assess those drugs into a more complex in vitro model that is the organoid. And those organoids can be pre-treated with the standard of care to mimic tumor recurrence. So the result of this work has been published recently in the AGMS in this special issue, and it will just walk quickly through the main findings. So in this screen pipeline, we screen for inhibitors for key potential targets for glioblastoma that we identify in a literature review in the field. These targets, there were 36 inhibitors in phase 2-4 clinical trials for glioblastoma, 20 inhibitors that were already FDA approved, and 15 inhibitors that were in development. So we used 2D cultures of patient-derived glioblastoma stem cells and did a high throughput screening. We performed around 4,000 tests that includes 67 drugs in a dose concentration manner for replicates and using 2 patient-derived glioblastoma stem cell lines. The main findings of this is that the response wasn't homogeneous across the 2 samples. Specifically, what we found that there were a group of drugs that killed basically both cell lines, but there were also a group of drugs that were specific for one or the other cell line. This again shows the role that plays the glioblastoma tumor heterogeneity in response to the treatment. When we look to what are the targets of these drugs for each of the patient-derived cell lines, we found that those targets don't overlap. So again, reaffirming that patient tumors are not equally sensitive to the same drugs, right? So with this identified drugs, then we, for this patient, because we also have a patient-derived glioblastoma organ, and we tested the effect of these drugs, and we found that, for example, costumelide was a drug that was good in affecting the viability of glioblastoma organoids in 3D culture, right? For both cases, for a primary model of tumor, and also for a recurrent model where the organ was first treated with chemo and radiotherapy, and then follow up with a treatment with costumelide. Costumelide is a third inhibitor, and just a reminder for you that third promoter mutations is affected in occur in more than 90% of glioblastomas. So this shows us that the model could be a good model to test the effect of these drugs, eventually something that can be later tested in vivo models. So one of the challenges that we are now looking to, trying to understand more about the intratumoral heterogeneity in GBM and the cell behavior that non-malignant cells play in the tumor microenvironment is our capacity to watch them in vivo. So we've been working on developing high content imaging approaches where we can look the behavior of these individual cells. We develop a microplate insert that is 3D printed, that it looks like an insert that goes into a multi-well plate. Basically, this cone, the function of this cone is to hold the organoid in close position to the bottom of the multi-well plate so we can do higher resolution imaging. This work also has been recently published in BMC Biomedical Engineering. And the advantage of this microplate insert is that allows to watch the organoids development during a long period of time, even up to 2 months. We also been able to do movies that are that long, and the use of these inserts then doesn't affect the viability. So these experiments were done using brain organoids as a test, proof of principle, on which we also perform more high resolution imaging. So these are different types of imaging modalities that correspond confocal light, confocal clear images where the organoid was clear. This is a multiphoton and clear organoid, and this is multiphoton and high resolution, where we can really see now individual cells within the organoid. We've also been able to do live movies. Here is, again, it's a brain organoid where we can see cell movement at high resolution within the organoid for up to 2 days in period of time with a very high imaging frame rate. And we apply this technology to also visualize the behavior, in this case, of cancer cells within the organoid. So here are cancer cells where patient-derived cancer cells, glioblastoma cells were labeled with the fluorescent marker and then introduced into the organoid. And then we can, now using this technology, we can really see the individual behavior, and our goal later is to start to assess the effect of drugs on cell proliferation, invasion, and also cell death within the organoid. That happening without affecting the surrounding healthy brain tissue. So with that, I would say that our lab is combining these 2 approaches that correspond to the in-depth analysis of the patient-derived biopsies and preclinical models of glioblastoma that are used in the lab to test the functionality of new molecular targets that we identify. Our focus is to find new targets that stop tumor progression by inhibiting those interactions that occur between tumor cells and non-malignant cells. So I just like to the end. So I just would like to thank to all the different collaborators that we set up for these projects and our funding bodies. Okay. For this seminar, so what we will do is that people will start to put the questions and answers in the chat, and then we will answer those at the end of the 3 presentations. So now we will go with the next presentation that is by Maria Jose Gatas from Buenos Aires, Argentina. Maria Jose is a PhD student in the laboratory of Marina Simian in the Institute of Nanosystems in the National University of San Martin. Her work focuses on the new therapies for glioblastoma based on nanotechnology. Her target is a better understanding of the tumor microenvironment, particularly tumor-associated macrophages, that has a high relevance for drug delivery and also targeting this type of heterogeneous tumors. So with that, I will pass the presentation to you, Majo.
Okay, thank you very much, Guillermo, for inviting me and introducing me. So I'm gonna talk about our recent published article called A Heterotypic Tridimensional Model to Study the Interaction of Macrophages and Glioblastoma in Vitro. So a brief introduction of glioblastoma multiforme. As Guillermo told before, glioblastoma multiforme is the most common primary brain tumor that constitutes about 70% of all gliomas in adults, and current treatments are based on surgery followed by radio and chemotherapy. It doesn't exist a specific treatment for glioblastoma nowadays. And survival time for glioblastoma patients is between 12 to 14 months. So why do we think that it's important to develop complex models? So we have presence of other non-malignant cells within the tumor, such as vascular and stroma cells, microglia, macrophages, and other immune cells. Here we have a brief summary of the glioblastoma tumor microenvironment. We can see we have innate immune systems such as macrophages. Also, we have adaptive immune systems, T cells, lymphocytes, dendritic cells, and all of them contribute in tumor growth and promote a tumor-promoting microenvironment. So this is why it's important for us to study also the microenvironment that is also part of the tumor for design new therapies. So we have focused this work in tumor-associated macrophages, also known as TAMs, and we can classify them in two different groups. So we have, again, microglia, tumor-resident macrophages coming from Yolk-Sac lineage, and we have macrophages that are circulating monocytes from bone marrow. They arrive the tumor as naive monocytes, and they mature into macrophages when they arrive the tumor. There's a constant crosstalk between the tumor-associated between these tumor-associated macrophages and glioblastoma tumor cells. So broadly speaking, we can differentiate these two into two groups. The macrophages, we have the M1 pro-inflammatory classical phenotype that we can recognize them because they are high in CD64, is a cell surface marker. And we also have the M2 pro-tumoral immunosuppressive phenotype that is high in CD206, CD163, and MERTK. These are typical hallmarks of M2 phenotype. So macrophages, tumor-associated macrophages constitutes about the 30 to 40% of the tumor mass, and actually M2 immunosuppressive phenotype is about 80% of all macrophages. They have several immunosuppressive functions that helps the tumor growth. Here I put an example, for example, that TAMS helps glioma cells to inactivate or to inhibit the activation of T cells. So our model, we develop a model to study these interactions. We use a human GBM cells, the U87 cell line, and we did two different experiments using human primary monocytes coming from blood donors. And also we used the THP1 monocyte-like cell line. And both of them, we did a 2D model using U87 conditioned media and a 3D model using U87 spheroids in co-culture with human primary monocytes or THP1 cells. So our results. What we did the first time was isolate human primary monocytes that usually are grown with fresh RPMI. This is the typical medium for monocytes. And we did one more control and our treatment that was adding RPMI and 50% of DMM low glucose, because DMM low glucose is the medium that U87 cells commonly use. And our treatment is monocytes using RPMI and 50% of U87 conditioned media. So we analyzed by flow cytometry CD64, that is a M1 marker, and CD206, that is a M2 marker. And what we could see is that there was an increase of double positive cells, CD64, CD206, in cells that were gated in CD11B. CD11B is a marker of macrophages in general. So we did gating in this population, and we saw that there was an increase of these two markers. And then when we used conditioned media here in green. And then we analyzed in also in CD14, that is a monocyte marker, only CD206, that is the M2 marker. And we also saw a significant difference between our treatment and one of our principal controls. So for us, this was very interesting because we thought that maybe we have soluble factors in conditioned media of U87 that makes these phenotypes to shift to the M2 type of macrophages. So we next question about the expression of mitochondrial P32. P32 is a protein very common in mitochondrial that assess the potential membrane of mitochondria and correlation in mitochondria. And it was previous bibliography says that its expression in membrane increases in tumoral cells and activated macrophages. So we look with confocal microscopy if this happens with this model of human primary monocytes in changing the medium 50% of DMM low glucose or 50% of conditioned media. And we could see an increase of the, well, here we have the green antibody is anti-P32 protein. And we could see that there was a significant difference between these two treatments and our controlled RPMI alone. So then we passed to analyze the THP1 cells as a monocyte and then macrophage model. THP1 cells are easier, obviously, to work with because we don't have to isolate them from blood every time we want to do an experiment. So we did similar experiments. We did first THP1 with RPMI, that is a typical medium. THP1 with PMA. PMA is some standard protocols use PMA to activate THP1 to mature them to macrophages. Then we did THP1 with U87 conditioned media and THP1 with U87 conditioned media and PMA to see if there was any stimulus to make them differentiate to M2 phenotype. So we analyzed again CD64, M1 marker, CD206, M2 marker, and we didn't see any differences in our treatments. Then we assessed, we analyzed CD163, that it was another M2 marker. And as you can see, we didn't see any difference also. And P32 in the membrane, we can see that we have expression of P32, but there's no differences between any of these treatments. So we passed to a 3D model. We wanted to make a co-culture of U87 spheroids with the monocytes. So we did U87 spheroids with a hanging drop method. We left them grow for 72 hours Here I'm showing just an image of a UAG7 spheroid. And then what we did was to add to this culture monocytes, or human primary monocytes, or THP1. Previously, we dyed them with a green dye to differentiate them from the spheroid. And here we can see in green, we have the human primary monocytes infiltrating the UAG7 spheroids. And also we saw the same with the THP1 cells. So both of them are capable of infiltrating spheroids, UAG7 spheroids. So then we next wonder if this infiltration makes any differences in the spheroid's growth or modifies its shape. So we track the spheroids for 20 days. And we saw, for example, these are spheroids without monocytes. We can see that they still being round at day 15. But when we did this with the UAG7 spheroids that were in co-culture with the human primary monocytes, we can see that there's a loss of sphericity. Here sphericity is maintained, and here we have sphericity lost. And we quantify them for more or less 10 spheroids per treatment, and we can see there's differences in the sphericity. And we are interested in sphericity loss because sphericity loss also is related with tumor malignancy and also metastasis. We assess the same experiment for THP1 cells, but we didn't see any differences in their sphericity. We see spheroids at day 15 that were in co-culture with THP1 cells didn't lose their sphericity. So finally, we wanted to characterize these infiltrating monocytes in the glioblastoids. Here I'm gonna show a little bit the gating we did with for the flow cytometry assays. We disintegrate the spheroids in co-culture with the monocytes, and here we can see, for example, this population of CD11 and CD14 low is a day 7 spheroid mark. Basically, these ones are the tumoral cells. Here, the ones that are CD11 positive, CD14 positive are macrophages, day 7 monocyte-derived macrophages grown in their natural media. So when we ask the co-culture, we can see that we have the day 7 spheroids. Also with two populations of monocytes, we have the one that we were expecting, CD11, CD14, and we have a second population here that we have CD11 positive, but CD14 low. So doing gating first in these ones, CD11 positive, CD14 high, we analyze the M2 markers, and we can see that the three of them are increased in this population of monocytes infiltrating the spheroid. Here, the light blue and pink curve are monocytes inside the spheroids, and we can see that there's significant differences between these monocytes and the control monocytes that are inside the spheroid at day 7, and with increase of the expression of CD206, CD163, and MERTK. We did the same analysis with the second population, that is CD11B positive and CD14 low, and we didn't see any differences in the expression of neither of the three markers. So we think that might be a second population of macrophages. And finally, we did the same with THP1, but as we were expecting, THP1 that were grown inside the spheroids didn't express any of these three M2 markers. So our discussion or conclusions about this work is that Fortunately, we could set a heterotypic 3D culture system using the U87 spheroids together with CD14 human primary or THP1 cells. Both of them are capable of infiltrating spheroids, but only primary monocytes acquire M2 phenotype as determined by cell surface markers and also in the impact of the sphericity of the culture and also in the 2D model using conditioned media. So our aim that was to develop a model to be simple and physiological relevant system to study macrophages tumoral cells interaction, can be used as a platform for preclinical drug testings. So I just want to thankful our group, Nanobiology Laboratory, whose head is Dr. Marina Simian, also is my supervisor of my PhD thesis. We work, as Guillermo told you, at the National University of San Martin in Argentina. And also this work, I want to mention, was done in active collaboration with the group of Antonio Carrera Silva and Dr. Andrea Errasti, that both of them work at the Faculty of Medicine and at the National Academy of Medicine. And we have to say thank you very much also to the people who, institutions that finance our work, the Evangelico Dodge Jack Yale Foundation, Grupo Dia, Agencia, and CONICET. So thank you very much for listening to my talk.
Thank you very much, Maria Jose. That was a very nice presentation. Okay, so I will introduce the next speaker. Is Professor Brian Day. He's a group leader at Queensland Institute for Medical Research and heads the Sid Faithfull Brain Cancer Laboratory there. Professor Day is an internationally recognized expert in brain cancer. He's co-director of the Center for Child and Adolescent Brain Cancer Research, and also he is a past ASM director and scientific advisory committee for the cooperative triage group for neuro-oncology Cogno, which is one of the most important here in Australia. So within Professor Day's achievements are the characterization of the receptor EphA3 as the therapeutic target for GBM, and also the development of a brain tumor and cell culture bank that sits at QMR Berghofer, which has collected more than 350 tumor specimens today and still collecting. So I'm very pleased to have Brian here in this webinar. I will just hand over to you, Brian. Thanks very much.
Thanks, Guillermo, and thanks to the organisers for the opportunity to speak. Morning, everybody, or afternoon, wherever you are. Yeah, and so as Guillermo was saying, I head the Sid Faithfull Brain Cancer Lab at QIMR in Brisbane. And really, I guess the focus of the lab is both on adult and paediatric brain cancer. You know, we're interested in basic biology of the disease, but you know, we're really, I guess, primarily focused on trying to stay as close to the clinic as we can, particularly interested in translational studies. So I thought I'd spend just a few minutes updating on what's been happening in the brain cancer space internationally. And so I would say really that the last sort of few years has been, you know, quite interesting. And this, I guess, really started politically driven. And so many of you remember, hopefully, Senator John McCain, who's a well-known American senator. Unfortunately, he was diagnosed with glioblastoma in July 2017, and he passed away fairly rapidly in August 2018. And then also, Beau Biden, who's the son of Joe Biden, and he was, I guess, this very well-known Attorney General of Delaware. And he was this, I guess, sort of very young, very popular character. And he was really struck down with brain cancer in 2013, passed away rapidly in 2015. And so out of that, I think, you know, really there was political impetus in the US and started this Beau Biden Cancer Moonshot program. And so you can see there on the left, they're injecting some 1.8 billion, really largely focused on brain cancer. And so I think that's really been a game changer and starting to really push the focus of brain cancer globally. And then here in Australia, there were political things occurring as well. And so one of our senators, Senator Katrina Billick, she was diagnosed with 2 brain cancers. And fortunately, they were benign brain cancers, and she's currently doing well. But she really went on, I guess, a mission as well to try and raise awareness and funding, and started the Senate Select Committee meeting on cancers with low survival rates in 2017. And I was part of this process. I was able to speak in front of this panel, and they really went around the country and tried to find out what the problem was. And so out of that has come this Australian Brain Cancer Mission. And the idea there is really obviously to try and make gains for the quality of life for people with brain cancer, and obviously improve survival. And so the budget there is really committing approximately $50 million over the next 10 years. And so a lot of that funding initially has gone into clinical trials. Unfortunately, no funding has gone into basic research yet. We're hopeful that that'll progress soon. And so I think you start to get a sense that, I think the community as a whole really has put brain cancer in the too hard basket for a long time. And now I think this funding and raised awareness, I think really augurs well for the future. And so big things also been happening in Brisbane in the kids' brain cancer space. And so the Children's Hospital Foundation met a number of years ago now and really wanted to find out what was one of the bigger problems for cancers with children. And it really turns out that, aside from accidents, brain cancer is really one of the biggest killers of children. And so that's really not well known. It's definitely the biggest cancer killer of children. And so they really wanted to start this Children's Brain Cancer Centre. And the goal here really was to improve every aspect of brain cancer that matters to children and their families. And so really, I think, once again, taking big steps here to try and, form these diverse collaboratives. And really we're interested in not only just basic research, but clinical trials and also survivorship. And so 5 million was committed to that initially anyway, and launched in 2019. This is just a visual of our, I guess, official photo. We were launching March 2019. And this is our unofficial photo where, I guess we're having a bit of fun. And so it's exciting, I think, to see these diverse centres get off the ground, and hopefully we can make improvements for these kids. And so I thought I'd talk a little bit, I guess, about, the problems of brain cancer. And so, I think there's some very clear disease obstacles which are holding back the field. And so, I've been in brain cancer space now for almost 20 years. And, I think finally the message is becoming clearer that really the current strategies are not working. If we keep doing what we've previously been doing, if the field keeps doing that, we're really not going to, move the needle. And so, we need to actually do better. And so there's been a few shining lights, really that we thought would work. And so one of them was this Depatux-M, which is an antibody drug conjugate against EGFR. And so initially it, had a quite a good signal in the recurrent disease, but unfortunately it failed in upfront disease, and that international trial was stopped. And then I think another hope was, immuno-oncology, and the hope of checkpoint inhibitors had worked in so many other diseases, other cancers, that, there was a hope that it may work in brain cancer. And unfortunately, it didn't really work initially. But now there's these neoadjuvant IO strategies. And so the neoadjuvant approach really is where you want to give your immuno-oncology before you resect the tumour. And so the idea there, I guess, is that the tumour really acts as a sink of not only immune infiltrate, but also the tumour cells that you can raise a response against, and you get migration of tumour cell of immune cells in and out of the tumour. And so if you give these checkpoint inhibitors 1 to 2 weeks before the patient's initial surgery, then you get a much better response. And so I think if I was predicting what might come in the near future. I know there's some large international trials getting going on neoadjuvant IO approaches. And so I think that's something that will come definitely in the future. And obviously the blood-brain barrier is still a huge problem. And so I think the field has got obviously so addicted to wanting drugs to work in the vein. And we know that's driven by large pharmaceutical companies. But largely we have this blood-brain barrier problem where we know we're really not getting enough drug into these tumours, in sufficient quantities to be effective. And so I think there's a couple of strategies that are coming. And so convection enhanced delivery is a fancy name, but in essence, really, it's intrathecal delivery of drug directly into the tumour. And so it's a highly specialised technique. But, this has been hampered really by the fact that these catheter tips really didn't diffuse drug in very well into the tumours. And so the technological advancements now have been around really, improving drug perfusion and these catheter tips. And so I can see now that there's some centres, Duke in the US and in New York, and also places in Europe and Japan, where they're starting to open up this convection enhanced delivery. Now, these are small trials, but they're actually getting very, very good results. And so a way forward into the future might be that we have specialist centres where patients can come access CED. And what I love about this CED approach is it really puts all the drugs that have ever been trialled in the vein back on the table. And so I think that's a really, could be a game changer in the future. And another one which you may have heard of is focused ultrasound. And so this builds upon well-established technology that they use in the heart for cardiac. And so the idea here is you infuse the patient with microbubbles and then pass an ultrasound over their heart, and it expands the blood vessels. And so people have now started validating this technology in the brain. And there's some very sophisticated work going on in animal models and also in humans, where it seems that you can use this technology to open the blood-brain barrier, at least for 1 to 2 hours, it seems, and then you can chase it with drug. And so once again, I think, we really want as many tools in the toolkit as we can. And I think CED and focused ultrasound are the way forward in the future, at least in specialist centres. And I think I wanted to touch also just on our tumour bank. And so it's something that, we've been developing here at QIMR for many, many years. As Guillermo alluded to at the start, it's not a huge bank, but we're up to sort of 350 odd specimens now. And really, when we started, we didn't just want to put a piece of tissue in the freezer. We really wanted to collect as much as we could in a detailed and sophisticated way. And that has really, I guess, been the foundation and building blocks of what we do, not only in my group, but many groups around the country as well. And so from that, I think we realised, Brett Stringer and myself realised a number of years ago now that really there wasn't a number of good cell line models to use. And so back then, people were trying with serum growing cultures, and really it was very difficult to get these things growing. And then we adopted a technique where we were essentially using laminin or matrigel, and then a serum-free environment with growth factors. And that was really a game changer for us in that at that moment, we started to be able to generate a cell line from these patient tissues very faithfully, probably 95% of the time. And so Brett and I went on and generated quite a number of these cell lines. And so these are now available freely for the academic community. And so we've characterized them in very great detail. And so we made a panel of 12, and all that information is available online. And they really have been very, very well taken up around the world. And so this is sort of a recent distribution map, I guess, of our Q-cell models. And you can see now they've been distributed in the UK, Europe, in Asia, many, many labs in Australia, New Zealand, and also in the US. And so I think they're in some 45 publications and leading papers like Cancer Cell, Nature Neuroscience, and others. And so we're all aware, I guess, here today of the revolution in organoids. And so I think organoids really, hopefully will fill that gap of what we're really wanting, which is a model which better represents the true disease state. And so here on the left, this is a typical 2D culture. So this is a photo of one of our Q-cell models where we're growing them on matrigel. And there's still heterogeneous populations, but really you don't have that 3D cell-to-cell contact that you would hope. Here on the right, we've taken this as a pediatric DIPG primary cell line that we're now growing as organoids. And hopefully, these are touted as better representations of disease. These models stain very floridly here for Nestin, GFAP, which is a glial marker showing more differentiation, and SOX2, which is a stem cell marker. And so you can see that we're getting nice mixed populations in these cultures as well. And so hopefully this video is working and you can all see it. And so really in my lab, Ulrich has spearheaded our organoid program. He's a very talented scientist and has really put a lot of drive into developing these models. And so here, this is a pure GBM cell line model here where we're growing them between two scaffolds, actually based on a cardiac model. And we've labeled two different cell populations, green and red, where we expect to see contact repulsion. And so hopefully you can appreciate that that is indeed what's occurring. And so these are obviously very unique models, and you can really start to assess some of these receptor-ligand interactions. The other work that Ulrich has started to get interested in is co-culture, and that was mentioned here by others today as well. And so, we've started doing some investigations where we're mixing, we've got normal neuronal cells, microglia, and also potentially blood vessels or endothelial cells with GBM cells. And here's just an interesting picture where Ulrich was able to show when we mix microglia in with GBM cell populations that we got this phenomenon where the microglia seem to actually corral and encircle the GBM cells and really preventing their spread. So, I think these are really just the dawn of these sorts of platforms, and the sky's the limit. And then, as Guillermo also showed a little bit of, I think these glioblastoma explant organoids really are the way of the future. They're very exciting. And obviously the idea here is that instead of enzymatic dissociation of cells into single cells, you essentially just physically chop them up and really maintaining all that heterogeneity intact. And then you're able to culture these in spinner bottles, I guess, in situ. And, you can really take these out, I think, even for 2 to 3 months. And so here's a nice example where, we have sort of small cultures forming at 2 days, at 8 days, they're rounding off. And then these quite large GBOs are forming. Here's some just some H&E images where we've taken sections of these GBOs. And really what we're looking for is, can we see this heterogeneity within these GBOs? And so here we're comparing the original patient tumor to the GBO. And so far it's looking, it's looking quite good in that a lot of the morphology we'd expect to see is there. And then Ulrich has actually gone further now and starting to stain for some of these elements. And so here you can see he's performed some immunofluorescence looking at Nestin, which is the stem cell marker, GFAP, which is a glial differentiation marker, and KR67. And so we're getting nice mixed populations. Once again, here he's showing CD45, SOX2. So we still have blood lineage cells in here and stem cells. And over here he's stained for CD31. And so the blood vessels really still seem to be intact. And IBA1, which is a specific microglia marker, which are the resident macrophages in the brain. And so these seem to be retained within these GBOs as well. This is just a little bit more of a close-up here. And so you can see you get nice proliferating cells throughout these GBOs and blood vessels. And so you get really nice, clear mixed populations of blood vessels. And amazingly, these blood vessels are able to be retained within these GBOs. I think it speaks really to the ability of these heterogeneous populations to work collaboratively to keep themselves all going. And once again, just showing that these immune cells here are retained. And so CD68, which is a marker of circulating macrophages. And so we now know within glioblastoma tumors, you get two populations really. You have the resident macrophage, which is microglia, but you also have bone marrow derived circulating macrophages, which are also transiting in and out of these tumours. And so we're able to pick those up within these GBOs. And so I think one of the challenges, as many of you be aware, is, you know, I think the real positive of a 2D culture is you can do platform screening. You can take you know, large numbers of drugs and you can put them through multi-well systems and get results that way. And so I think that's still quite a valid approach, where you might use your 2D cultures to do pre-screening, identify drugs of interest, and then, you know, you want to take your higher end organoids and GBO platforms, which are a bit more labour intensive, and actually use those to take the validation further. And so I think what we're starting now in my group is to really just work out how we can get good information. And it was really nice to see Guillermo's data as well on how he's doing that. And so here what we've done is just treat with a very common, well-known tyrosine kinase inhibitor, dasatinib. Post-treatment, we're sectioning these GBOs and then staining for cleaved caspase 3, which is a marker of apoptosis. And so these are sort of more labour-intensive processes, but hopefully we're getting closer to what we would expect in the real patient setting. Okay, so I thought I might change gears a little bit and just talk about my work on F receptors. And so I've had a long-standing interest, I guess, in EPH or F receptors. And I guess these are well known in the context of embryogenesis. They're broken into either FA or FB receptors, so named for their binding to either Ephrin A or Ephrin B ligands. We know they're widely implicated during embryogenesis, where they are involved in tissue boundary formation, organ development. This family of receptors, comes together and forms these tetrameric complexes, where you get not only forward signaling through the kinase, but also reverse signaling through the ligand. And so these were, I guess, early on widely implicated during embryogenesis, but it's now been known for a number of years that F receptors are re-expressed and functional in cancer. And so a particular family member, FA3, I got interested in a number of years ago. And why we got interested in that is that FA3, really appeared to be significantly elevated in approximately half of these GBM tumours compared to normal brain. And really importantly, it appeared that FA3 was acting as an oncogene. And so when we correlated gene expression with survival, what we found is that when FA3 was upregulated in these patient tumours, the patients did significantly worse, much worse, in fact. And when these patient tumours downregulated FA3, they did better. And so it gave us a strong, signal that FA3 could be implicated in tumorigenesis. And I won't go into detail on a large body of work that's been published, but really the summary is that we found that FA3 was expressed in adult GBM. It negatively correlated with survival. It was really, I guess, really particularly overexpressed in the most aggressive mesenchymal subtype, and later we've been able to show that it has a function in driving that. It was on the more stem cell-like population, and when we knocked it down, it delayed tumour formation, and we went on to look at targeting strategies of that. And so I wanted to just touch on some of our work in trying to translate those findings now. And so my long-term previous mentor, Andrew Boyd, teamed up with Andrew Scott and the late Martin Lackman many years ago now with this US-based company called Humanigen, and they took a mouse FA3 antibody and took it through this humaneering process and really started the steps to turning this into a therapy suitable for clinical trial. And out of that work came this so-named favetuzumab drug, which is I guess a recombinant antibody targeting FA3. And that initially went through studies in primates and then into phase 1 in leukemic patients. And so we had this platform now where we could take this drug and trial it in glioblastoma patients. And so we got some funding from Cure Brain Cancer Foundation, which was very generous to get this going. And so the trial has been led by my friend Hui Gan down in Melbourne and Andrew Scott at the ONJ. And then it was 2 sites, so really at Austin and RBWH. And so Paul Thomas, Zana Lewin, Poe Inglis, and Lindy Jeffrey have really been supporting this trial in Brisbane as well. And so the idea, it's a fairly small phase 1, a dozen patients or so, really a phase 1 safety and bioimaging trial. And so, I think that the really exciting part was we were able to take the drug and label it to zirconium-89, followed by PET imaging. And what we wanted to look for was brain penetrance. And so here you can see in this recurrent GBM's patient, you can see very clear delineated tumor here. One of the standard imaging modalities, FDG, here showing once again the tumor borders. And here when we use our anti-FA3 antibody labeled to zirconium-89, we're getting nice tumor uptake in this patient. And so I think this was, one of the really nice things about this trial is we're able to show that at least in recurrent glioblastoma patients that this antibody is accessing the tumor. And so there is, I guess, now becoming more established that in recurrent disease, more so there are parts of the tumor where the blood-brain barrier is open. Also, we had an inkling that FA3 might be enriched in recurrent disease. It seemed to be this tumor-specific marker, hopefully, and then also there was previous literature saying that, that mesenchymal subtype GBM is enriched in recurrence, and we knew that FA3 was part of this process, and so we expected that FA3 might be enriched in recurrent disease. And so Kazi et al. In 2018 was able to show that quite elegantly, that FA3 is indeed significantly enriched in recurrent GBM patients, and also it's elevated following chemoradiation. And so one of the things that we wanted to do in my group was really start validating antibody drug conjugates, because obviously these are aggressive tumors, and we want to hit them as hard as we can. And so this is really, I guess, an old technology now, where there's a number of different chemistries where you can link cytotoxic agents to antibodies, and that technology has now improved, and really you don't have that problem of, in essence, drug falling off and associated liver toxicities and things. And so here, I guess this is showing our antibody penetrating these GBM cells. So when we treat these cells, we get activation on the cell surface and internalization of receptor complexes, which is exactly what we want. And so this drug has been pulled into the cells. This work has been really driven on the ADC side by Chris Thorect at UQ, and he's really been developing these non-cleavable lysine attachment ADCs for us. And so does it work? This is a bit of data in the dish. And so we've taken two of our Q cell models, an FA3 positive one and an FA3 negative one. And indeed what we see is a nice dose increase, a dose response here with the ADC in an FA3 positive model. But when we treat an FA3 negative model, we seem to really have no effect. So that's comforting in that we seem to be getting quite specific targeting. And are we killing these cells? So yes, within sort of 24, 48 hours, we're seeing clear caspase-3 come up with cell death peaking around 72 hours. And then we've taken that through to animal models. And so here we've engrafted orthotopically. We've got 3 treatment groups: PBS, just the naked antibody, and then this FA3 antibody attached to USAN, which is a tubulin inhibitor. And so you can see we've allowed tumours to form for 3 weeks and then commence treatment. And we get a really quite a nice anti-tumour response. And we seem to be, not completely curing these mice, but we're essentially clearing a lot of these tumours. And despite that fact, interestingly, when we kept therapy going, what we found was that these tumours still recurred on a lag, and that led to really a doubling of survival of these animals. And so I thought I'd touch now, what we've moved on to is to start to understand why this mechanism might be occurring, where when you hit these cells with FA3, even though we continue to hit FA3, they're coming back. And so I think this really speaks now to this heterogeneity concept within these tumours, where when you target one population, unfortunately, it's the multi-headed snake. And so you're cutting only really one head off and then another head grows. And so we started to look at this ligand. We'd had a hint that potentially there might be some contact repulsion going on between this receptor and ligand. And so ephrin A5 is the high affinity ligand for FA3. And when we looked at expression, we found that ephrin A5 was really, I guess, expressed at comparable levels. And here's FA3. When we treated those mice that I showed you on the previous screen, when we looked at FA3 levels, as we expected, the FA3 levels were down. And so we were reducing the FA3 positive tumor cell population, which was comforting. That's what we would hope. But when we looked at the ephrin A5 levels in these ADC treated mice, we found, if anything, they were elevated or retained. And so when we now wanted to look at the expression of this receptor and ligand pair within these GBM tumors. And lo and behold, what we found was, I guess, this very, very discrete pattern of staining. So here we've taken a GBM sample where you're seeing sequential sections, and on the left, FA3 staining, and ephrin A5 on the right. And so hopefully you can appreciate that there is this very clear contact repulsion going on where you have very strong FA3 regions, but also very strong, clear ephrin A5 regions. And so, when we combine both of these together at the expression level, we quantitated that, and really we're getting target coverage reaching almost 80% of these tumors. And so the idea now is, well, can we target both of these? Can we hit them both at once? And as I alluded to earlier, these are aggressive tumors, and so I think your first punch is your strongest punch. And so we're now working up this idea in my group where we have novel antibodies against ephrin A5 and FA3, ADCs in development, almost about to take them in hand and start validating these dual targeting responses to really get a better coverage of this heterogeneity. And then I think another game changer for us has been this idea of cell states. And so I guess a number of years ago, Verhaak and colleagues, and even before him, started to define, I guess, subtypes of the disease. And so we understood that there was, different drivers, but also I think many of us in the field had this hint that potentially this was a dynamic process and there may be some interchangeability. And so Neftel and colleagues in Mario Suva's lab in Boston late in 2019 published this paper in Cell talking about these cell states. And really, I think it's, it's a better reflection and a better way to think about this heterogeneity where there seems to be definitely, multiple types. And so these so-named cell states, OPC, MPC, AC, and MES-like are there. But what they showed elegantly really is there appears to be, I guess, varying levels of these in particular patients, and that there's indeed interchangeability between these cell states. And so we've been thinking about that in the context of targeting. We know that FA3 is on this, this mesenchymal portion. So we're now trying to validate where this ligand fits in the model. But, I think many of us in the field have this strong sense that if we're going to get durable responses, we really need to get a better handle on this heterogeneity. How do we do that? Well, will multiple targeting strategies work? Another idea I quite like is this idea of differentiation. So can we push these four cell states, can we push these four cell states to be, through a differentiation strategy to be more one state, right? And then potentially slow the tumor down, or if we can push them all in one direction, then we can increase the chance of, single or dual targeting strategies. So I'll leave it there. I think we still have significant brain cancer disease obstacles. The blood-brain barrier is a major hurdle, but I think there is certainly ways and means coming into the future that we'll be able to hopefully circumvent some of those problems, particularly CED and focused ultrasound. I think GBOs are a really exciting, exceptional platform, particularly for studying heterogeneity, and hopefully will really speed up what we're doing, get us closer to the clinic. I'm personally in the lab, as I said, I think we're very focused on trying to define targets that are specific and targets that have a better coverage of heterogeneity. So a lot of people have gone into bringing this work together. So thank you to all my lab and my also key collaborators and funding bodies. Thank you.
Thank you very much, Brian. It was a very nice presentation. So I will check questions from the attendees.
I mean, Gigi, the only question sort of occurred to me perhaps to ask you was just around, I think one of the issues we're having is that with the 2D culture, I think sometimes when we put drugs on, we really don't see nice heterogeneous responses, at least not in our hands. Often, you know, we would hope for more selectivity, but often we don't see that. And I think that's one of these vagaries of the 2D culture where you're just getting heaps of drug contact and it's not very selective. And so I guess we're still just getting into doing, I guess, the drug therapy in these organoids now. And so I guess we're hoping for more heterogeneity. I think you're further along in that process than us with drug targeting. And so have you experienced more heterogeneity or varied responses in 2D versus 3D at all or not?
So I think what we are looking is to use, I think the starting point was this list of drugs, right? And then see how they target different cells from different patients, right? So we can look into the heterogeneous response within a single culture, right? See, for example, when we have 50% cell death, what is that, right? So is 50% of one population, or just by chance, 50% of cell got the drug? I think that needs more careful analysis. But
I think it's one
to choose the thing, right? But
I think it's one of the ideas that we've thought about is, I think it would be really nice of those 4 cell states to have a robust system where you can say, okay, well, drug X has been able to eliminate, just that population, or, it's got a better targeting. It's eliminating 80% of the OPC-like and, not really targeting the others. And so I think we're just starting to get into those thoughts about how we can probably through flow cytometry initially anyway, start to look at some of these targeting strategies within these GBOs and things.
I think, I think that everything is evolving, that everything has different states, right? Like once we get better insight about the markers of the state for tumor cells and non-tumor cells, right? Flow cytometry would be a good thing, right? For the organoids, because we need to do single cell suspension. So that is an easy, rapid way to quickly analyze. The 2D cultures might be suitable for a high content imaging approach, right? So where you just label with different markers and see what is left. What will be also required is that all those experiments done in a dose response manner, right? Because if you add too much of a drug, like as you mentioned, you might be killing everything, right?
Yes.
But you lose the specificity. And that is, when we analyze responses, we look at the IC50 thing, right, as an indicator, right, rather than just sitting in a high concentration part of the curve.
Yeah. No, absolutely.
Yeah. Okay. So Majo, for your system, I don't know if you have explored the ways just to see whether those M2 phenotype, right, of the human monocytes can be reversed. Have you seen whether you can reverse the phenotype? Basically you culturing in conditioned medium by tumor cells, and then you got the transition. But whether you put in a normal media, did you get them back?
No, we are trying to develop strategies. My principal aim of the PhD thesis is to reverse this phenotype to a M1 phenotype by targeting with specific peptides. But we are studying that now, so that's why I didn't show because they weren't part of the paper. And we are trying to target these macrophages, M2 macrophages, because we have peptides that target M2 macrophages. And the aim was to put a put nanoparticles with these peptides and try to send sirnas, for example, Dicer, that is one of the sirnas that it was reported that can reverse this phenotype as a specific therapy, right? So obviously monocytes are it is very plastic, the way they differentiate. And so we are trying to, so we have this model now to test if we have any differences. So this is we're trying to do this.
Okay, that's very good. Brian, I have a question from the chat, so I will read it. Does the GBO retain the heterogeneity observed in a patient tumor? If so, do you see a selective pressure on a particular subtype, such that with time, cell with other subtype characteristically eventually takes over, right?
Yeah. I mean, the answer is we don't know, but we suspect that might be the case, right? I mean, I think what we see is that obviously the tumor cells are there's a population of pluripotent tumor cells within these GBOs, and Guillermo will have seen this as well. Is that when you cut them up, like physically, they have very sharp edges, but then they seem to round off quite quickly. And so, we make the assumption, and we don't know for sure, but we make the assumption that what rounds them off is the tumor cells, right? It's probably not, it's probably not the stroma doing that, right? And so I think, given the growth potential of GBM, I think definitely like what's happening is that we're getting changed proportions of the tumor cells. And so I think over time, you probably get increasing populations of tumor cells within these GBOs. And, it would make logical sense that you are probably enriching for certain types of tumor cell, but we don't have that data.
Okay, thank you, Brian. Okay, so I don't think we have any more questions. So Majo, you have anything you want to ask or expand on your project? Then basically.
Yes, I have a question for Brian. I like very much the model where you include the microglia cells. I didn't understand very well if those microglia cells were you add them in the culture, right?
Yeah. So yeah, I think in that instance we have we're actually using HMC3s, which are they're whether you know that line or not, it's an immortalized human microglia line. And then the other setting, we have this Ren cell line, which is a commercial immortalized neuronal cell line, and then we've got our Q cell models. And so we just had the platform where we've got many different fluorescent markers that we could use. And we just really wanted to see if we could sort of make a mini brain, I guess, and how those different elements would start to mix. I didn't show the data, but we've done the Ren cells as well, and interestingly, there seems to be quite a lot of contact repulsion going on within the population. And so the microglia, as I showed, seem to encircle things, but when you put the neuronal cells in with the GBM cells, we couldn't get them to mix. They were kind of like oil and water, and so
okay.
I think what's going on is you've got many different like adhesion receptors, and so I think there was a strong signal between these.
I thought it was very interesting because it happens like the contrary of what happens in our model. When we add monocytes, we have spheroids spreads. A monocyte infiltrate and then makes the spheroids go like bigger, and also they lose the shape. And for me, it was like, okay, surprisingly, it happens like the contrary, like they make like a circle or something like that, and they don't leave them tumor cells, no? So I don't know if maybe they act like a protective way of the, immunoinflammation, maybe?
Absolutely, yeah. I mean, it was very interesting dynamics. I think the problem is that around the time we were doing that, this new GBO technique came out, And so then to be honest, we've sort of stopped working on the mixed populations because the GBOs seem to have all of those elements in situ anyway. And so it's probably GBOs is ultimately a better platform. I think if you're looking at particular elements that you want to modulate, knock down or overexpress genes, then I think that, those mixed models are interesting. I think if you're looking really for just drug response and targeting heterogeneity, blood vessels, immune cells, and so on, then the GBOs are really probably the way to go. Gigi, can I ask you a question?
Thank you very much.
Just a little bit about, so you're growing normal brain organoids, is that right?
Yeah, we also do that. Yeah, from the
Yeah.
iPS derived, yes.
Yeah, that's cool. And how long do you sort of grow them for?
So we basically using these microplate inserts, we reach 60 days or something like that. Yeah.
Okay.
Yeah. So
because I have seen presentations where people have grown them for 6 months and things, and you get lots of interesting structures forming.
Yeah. So basically, there was a recent paper from Mine and Lancaster, right? They push longer, and then they got all these source of cerebrospinal fluid, production within the organoid.
Wow.
And that is something very interesting, right? For
really good.
For the context of GBM and so on.
And so you showed those nice images of the tumor cells infiltrating.
Yeah.
I think that's a really elegant model, right? It's a really elegant way to look at drugs that stop migration and all those sorts of things.
Yeah, that is the aim. So as I mentioned, one of the things that we will start to look at is, we understand the heterogeneity, but sometimes you might need to pay particular attention to each cell compartment, right? So the movie shows the GBMs, but macro shows the macrophages, right? So we might go into start to looking how these individual cells interact, and segregate. So basically, gaining insight into the granularity of the problem is the key challenge now. Mm-hmm.
I think the other strength of your FDA drug screening would be, like, I think I've always had this idea where potentially if we could get a platform like GBOs that faithfully matched the patient response, then we could take, at time of resection pre-treatment, we could take those patients' tumors, we could grow GBOs, and then we could do screening for FDA approved drugs while the patient is going through standards of care. And actually try and work out what drug is going to respond best, obviously. I think it's a simple idea, but it's actually like quite difficult to pull off, technically, but also logistically making all that work, having that pipeline working. And so hopefully we're taking the first steps, I think, to kind of getting that on the way.
Yeah, I think, look, probably I think we're on the right steps, moving that direction. One of the things I think we need to do is to, we are doing a screen of 60 drugs, right? So we have that screen reduced to 12 drugs, right? That identify 90% of the responders, right? That would be a good step. And the other is have those 12 drugs, with some sort of high-end level of validation, right? So they will cross the blood-brain barrier, no side effects, or so more tailored and a small group of drugs.
But I mean, it would, as you say, a panel of 12 would still be a massive, that would be a game changer in terms of what you could offer. Yeah,
I think, So for now, obviously we're working together in having this panel of GBOs, right? Where we can start to identify those group of drugs that can stratify responses, right? So this group of patients, associated clinical history, respond to this particular group of drugs, this other one, and so on, right? And that can be the fuel for a trial, right? Depending on what drugs are being selected.
Yeah, absolutely.
Okay. So
Sorry, Guillermo, we have one more question.
Yes.
I have my collaborator, Dr. Antonio Carrera Silva, who's the last author of my paper also, together with Marina. He's watching this by YouTube, and he out we saw AXL tyrosine kinase receptor upregulated in U87 cells. I didn't show that. And he says, as you saw in some of the tumor samples, do you know about the role of the receptor in GBM?
Yeah.
In glioblastoma.
Sorry, I don't know. I'm sorry.
You don't?
No.
No.
But
okay.
I'd be interested to have a look.
Yeah. Of course, but well, maybe you know that. Okay.
Yeah. No particular in that knowledge, but I'm very sure. So the thing with tyrosine kinases in general, that the inhibitors, sometimes are not very highly specific. Maybe an antibody approach for the receptor could be more suitable rather than the inhibiting the function. But I'm no expert in the field.
Not sure. Yeah, because we did the analysis of the expression of AXL, that was one of the TAMS receptors, and we didn't see difference, or we didn't see so much expression in macrophages, but interestingly, we saw expression in the U87 cells when we were expecting the other way around. So that why Antonio is an immunologist. He's not an expert in glioma, but he's an expert in macrophages. So that's why he was asking about if the expert in glioma knows something about AXL. But okay.
Yeah, I think I think it's quite interesting, the macrophage phenomenon in GBM, where we obviously you have the resident macrophages, but you also have bone marrow derived. But I think that obviously there's this classic M1, M2 kind of thinking, and I think the field is probably still confused, around the roles.
Yes, it is. It is.
Yeah, of what, whether they're having an anti-tumor response or whether they're actually pro-tumorigenic. I mean, it seems in some cases that having a lot of microglia there is a bad thing. And so it seems that, maybe it's just through inflammation or whatever, you actually, the patient can do worse if you have the wrong macrophages there in large numbers. And so, I think we still haven't worked that out.
Okay, so I think with that, we just finished the webinar today. I will say thank you to Brian and Maria Jose for accepting the invitation and give such nice presentations and updates on their progress in research in GBM. I also would like to thank the IGMS for organizing the webinar, Satoshi and May for their support, organizing all links and things. So we'll be organizing next seminar in July approximately. Just keep watching this channel with more updates. And also this reminder, we have still open the special issue on glioblastoma heterogeneity and plasticity for those ones that can submit, would like to submit an article there. All right. With that, I would say thank you very much.
I would like to thank the chair and the speakers for their time and interesting contribution to this webinar. Also, a thank you to our attendees. I hope you found this webinar interesting. As a reminder, the recording of the webinar will be made available as soon as possible on the webinar's webpage. I wish everyone a nice day, morning, evening, wherever you are in the world. Bye-bye.
Thank you. See you all. Bye-bye.
See you guys.